Good afternoon. I would have loved to join the VLIZ conference today, but I'm currently working abroad with a project called Coral Assist. But now I'd like to talk to you a little bit about my master's thesis, and this was to test the effect of sewage-derived nutrients on coral reefs in Malaysia, specifically the island Pulau Redang. This island has experienced huge growth in tourism over the last 2 decades, and it's showing no sign of slowing anytime soon. From 2016 to 2017, the year leading up to this project, the annual number of visitors to Redang doubled, as you can see here in this graph. Tourism waste could be a big problem for Redang reefs in the future. The resorts on this island all lack sufficient sewage treatment facilities. They use individual seepage pits, which collect human waste and slowly filter this through the beach sand and into the sea. This nutrient enrichment has the potential to cause algal blooms and declines in coral coverage and coral reef health in general, which in and of itself is the main attraction for tourism in the first place. This project used a combined approach, using video transects to estimate the percentage cover of all benthic groups, and using nitrogen and carbon stable isotopes to determine nutrient sources measured in 3 common reef organisms. The algae, Lobophora, uptake nutrients very quickly, while Acropora and Drapella have more long-term nutrient assimilation periods. This project aims to assess the best bioindicator of sewage pollution, to determine the extent of sewage pollution at Redang, and to test the concurrent ecological implications. So which was the best bioindicator? The algae was no good. Delta-15N reproducibility was unacceptably low, and this is shown by very high within-sample variance, as you can see in the black bar on this graph. Drapella was also a poor bioindicator of nutrient sources, and you can see this on the nitrogen-carbon biplot, where nitrogen is on the y-axis. There was no difference in delta-15N between any region, likely due to trophic fractionations and variable diets of Drapella. However, Acropora was a great bioindicator, with significantly enriched nitrogen signals at the tourist hub, indicative of sewage-derived nutrient influence in this area. Important results from benthic video surveys were the presence of 4 distinct reef communities. High coral coverage, Acropora and Heliopora communities were found in most regions. However, in the tourist hub, both a highly diverse Porites-dominated community, which is a coral-dominated community, and a heavily degraded zoanthid-dominated community were found alongside each other. Zoanthids are a solitary soft coral that, as you can see in this lower picture, have entirely covered this coral rubble which was once a live and thriving coral reef. Combining the isotopic sewage evidence with coral reef community composition data led us to find interesting regional trends. At higher levels of sewage nutrient enrichment, there's a greater regional variance in ecosystem community structure. This is characterized by the coexistence of degraded and non-degraded reefs alongside each other, only at the tourist hub. So to conclude, Acropora would be the best bioindicator of sewage nutrients in Redang, since it picked up a strong signal of delta-15 nitrogen in this case study. And we suggest that there's regular isotopic ecological monitoring from this point out. Secondly, the sewage treatment systems on the island need improvement. The current seepage pit system does not properly treat sewage, and a tertiary treatment system that's shared between all the resorts should be put in place. And then finally, there needs to be some better cooperation between government sectors. So currently, the land-based development is run by the state government, whereas the marine jurisdiction is under the Marine Parks Department. There's often a disconnect between these 2 agencies, and we suggest closer collaboration between the 2 in order to make development decisions which suit both tourism objectives and marine conservation objectives. Thanks very much, and although I can't take any questions, here's my email, so if anyone would like to email me about my project, feel free. And a great thanks to all the people involved in this project.